Thank you, Nick. So I'm going to talk about spectral editing at 3 Tesla and 7 Tesla, and after that I'll talk about drawbacks and how do we solve those drawbacks, and finally I'll mention about faster editing. So a brief overview of my talk. So I'll first start with, as I mentioned earlier, why do we need spectral editing, and how to perform spectral editing, and what are the variations in GABA measurement based on the recent multi-site data that we have acquired. And then after that, I'll talk about universal sequences, how did we standardize them as well, and why do we need to standardize them across all MR vendors. What are the drawbacks of MegaPress as a single metabolite editing method. And finally, I'll talk about the faster editing using Hadamard encoding at 3 and 7 Tesla. So starting with why do we need spectral editing. Over here we have a proton MR spectrum acquired using proton magnetic resonance spectroscopy. It's a proton magnetic resonance spectroscopy. It's a widely used technique to measure endogenous metabolites such as NAA, creatine, choline. However, due to limited dispersion along the chemical shift axis and broad line widths, which is largely the problem at 3 Tesla, large signals often overlap with small ones. So for example, over here, creatine in red overlaps with GABA in green. And this leads to ambiguous measurements of low concentration metabolites such as GABA, glutathione. So how do we solve this problem? We apply J-difference editing. It's a method that reduces the amount of information on the MR spectrum, removes the overlapping signal, and reveals the underlying signal of interest. At 3 Tesla, we use MegaPress to perform J-difference editing. It's largely based on press for voxel localization. And press voxel localization, you can see it's acquired by having a press sequence with slice selective excitation pulse in one direction, and then a pair of refocusing pulses, slice selective refocusing pulses in perpendicular to each other, and the excitation pulse. And when you incorporate a pair of editing pulses, you can now perform mega editing. So what are the metabolites that we edit, usually using this sequence? First one is the most common one, which is GABA. GABA is the main inhibitory neurotransmitter. You can apply that editing pulse at 1.9 ppm and detect it at 3 ppm. Glutathione is a redox compound. It's a marker of oxidative stress. Can be measured by applying editing pulse at 4.56 ppm and detecting at 2.95 ppm. And then we have lactate, which is anaerobic product. Can be measured by applying editing pulse at 4.1 ppm and detecting at 1.3 ppm. You have NAAG, a neuromodulator, measured by applying editing pulse at 4.6 ppm, detecting at 2.7 ppm. And we have 2HG, it's an on-call metabolite, tells you the IDH mutation status in tumors. It can be measured by applying editing pulse at 1.9 ppm and detecting at 4 ppm. So the next slides will be, I will focus on GABA as the main metabolite for editing, and then I will also touch on lactate. So how do we perform MegaPress editing? It largely involves 2 steps. The first step is where you do not apply editing pulse, hence the edit off step. And then the second step is you're applying editing pulse during the localization sequence. So over here, you start with the edit off step. You do not apply any editing pulse. You end up getting a conventional MR spectrum. In the second step, you apply the editing pulse. And over here, you apply at 1.9 ppm. Because of its closeness to NAA in frequency, you end up saturating the NAA signal. And you can repeat this n number of times, usually 320 times, to get sufficient signal to noise ratio. And now you take all the ons for GABA minus all the offs for GABA, and you get your GABA edited spectrum. And one point to note that is that the editing pulses do not affect your creatine signal, as mentioned earlier by de Graaf, and that results in removing your overlapping creatine signal and revealing the underlying GABA signal. So we have this sequence available across all vendors. However, they have diverse implementation of MegaPress sequence. So the next step is to standardize across this sequence across all vendors. But before I go into why do we need to do that, I have a question for you all. Hope it works. Oh, look at that, it works, and somebody answered very quickly. Okay. So what are the MR vendors do you use to conduct MR experiments? What do you use? Do you use Siemens, Philips, GE, Canon? Oh, thank God, everyone uses something. One of them at least. So you can see different sites have scanners from different MR vendors, and this gives us motivation to have one sequence across all MR vendors, or at least a sequence that is standardized across all MR vendors, so that we can perform comparable multi-site studies. And so looking into the background now. So several studies have conducted reproducibility in GABA editing, and they found that the coefficient of variation varies between 5 to 25%, depending on the region that you scan, whether you're doing frontal, occipital, parietal, and depending on the protocol that you use, whether it's site customized or vendor provided protocol. And between site and vendor differences in CV are less well understood, and therefore it's very important to have the sequences standardized. Why? Because it will increase usefulness of the literature and also positively impact multi-site studies. To look more into this variation, we recently performed multi-site study on GABA editing. And you can see here there were 24 research sites involved around the world, and some of them had Siemens with blue dots shown on the screen, and the ones with Philips are orange, ones with GE are green. And this paper is published by my colleague Mark Mickelson. You can refer to the paper, what are the protocols involved, what are the differences between protocols, and the qualitative and quantitative analysis of the data. However, I'm more interested in this particular data, which is GABA to creatine results. And over here, we can see that data from GE are presented as green boxes, Philips with orange, and Siemens with blue. And you can see that there's overall agreement across sites and across vendor. Later on, you can see that. However, there's also variation that we need to look into. And we can see that within vendor, that is within Philips, Siemens, and within GE, you have variation as well. That's about 11.5% for Philips and GE, and slightly lower for Siemens. If you combine the whole data set, you have about 12% variation. And breaking down this data set, you can see that 8.2% was attributed to vendor level differences, and about 20% attributed to site level differences. And then the rest is not contributed to either of them. So combining that, about 30%, you have differences attributed to site and vendor level differences. So why is that? So if you look at here, these are the protocols that are implemented for that multi-site data. And you can see that there are differences across vendors in how the megapress sequence works, and you have differences in RF waveform used for the pulses, site selective pulses. You have asymmetric pulse for Philips, while most of them use Philips and Canon have asymmetric pulses. Also for refocusing pulses are asymmetric for Philips and Canon. However, then they use symmetric for Siemens and GE. The editing pulses are different in terms of waveform, but it also different how far apart they're kept, and that affects your editing as well. And in terms of timing, TE1 and TE2 are also different across vendors. And these differences lead to edited signal with different patterns, and that also can cause variation in your measurement. So therefore, that's our objective was to design a sequence that's a megapress sequence with standardized RF pulse length, shape, and bandwidth, and also the timings. So how did we do that? We started with a press sequence with editing pulses in it. So we have asymmetric excitation pulse for slight selective excitation pulse, and we have a pair of slight selective refocusing pulses. They were all optimized across all vendors, and also editing pulses which are sync Gaussian. In terms of TE1 and TE2, the timings, the TE1 was fixed at 13.1 milliseconds. That is independent of echo time, and then your TE2 varies with echo time. And by definition, your refocusing pulses are half of echo time apart, and your editing pulses were maintained half of echo time apart. So let's look at the results. We implemented this sequence in Siemens, Philips, GE, and Canon. We're starting with Siemens and Philips. So we had 6 subjects, and we tested across Philips and Siemens, and we overlapped their data for GABA editing. And you can see that in terms of signal line width pattern, they agree across vendors. We performed these scans in additional 2 subjects, GE and Canon, and then we overlapped the data over a variation that is from the combined Siemens and Philips data. And you can see that the data looks excellent and it falls within the variation. And then we did lactate editing at longer echo time. And again, we get excellent data and excellent agreement across vendors. And again, GE and Canon gave us excellent results as well, comparable across all vendors. We quantified the data and also looked into how much does it vary. So starting from vendor native, that is a vendor provided protocol, the variation was about 7%, that reduced to 3% using universal sequence. And lactate editing, there was a remarkable improvement, that is from 41% to 8%. And this is mostly because some of the vendors, such as Siemens and Canon, they have editing pulses which are not half of echo time apart, so that affects your refocusing of your coupling evolution. So So we have this megapress sequence. It works across all MR vendors, very reliable. They can give you the metabolites that you want to measure. However, the drawbacks associated with this sequence, and first and foremost, is that it selectively edits one metabolite, one target metabolite in one brain region. So you can perform this editing of GABA or lactate or glutathione only, so that you focus on one metabolite only. And then because of the small signal intensity, you require long acquisition times for sufficient signal-to-noise ratio. So for example, if voxel size is about 25 mils, you're looking into if you acquire on 300 averages, you're looking into a scan time of about 10 minutes at a TR of 2 seconds. And this is just for one metabolite. So if you have multiple metabolites, you're looking in multiples of 10 minutes. So the question comes in, we have a given time frame. Can we acquire more data? Can we speed editing? So I know that just to recap, so I'll still focus on GABA, but now I'm going to jump on glutathione as well. So in order to answer the question, how do we perform faster editing, we need to go back into our editing strategy using megapress sequence. So over here, we have editing performed using interleaving on-off steps. And you can repeat that about 300 times to get glutathione's edited spectrum with sufficient signal-to-noise ratio. However, over here, on step is where you apply the editing pulse at 4.56 ppm. And then you can repeat the same steps for GABA editing. And the order is not crucial. Turns out you can swap these two, and then instead of performing interleaving on-off, you now perform pairwise interleaving on and off, and you can repeat again that 300 times and get your GABA edited spectrum. The on step is where you apply editing pulses at 1.9 ppm for GABA editing. Putting these two editing strategies together, now you can see that instead of performing two-step editing experiment for one metabolite, you can now perform four-step editing experiment for two metabolites at the same time. And now we're going to call them experiments A, B, C, and D. Experiment D, you don't apply any editing pulse. And experiment C, apply the editing pulse for glutathione editing at 4.56 ppm. Sorry, can I go back? Anyway, it's fine. I'll just... So experiment C, you apply the editing pulse at 4.56 ppm for glutathione editing. Experiment B, apply editing pulse at 1.9 for GABA editing. And experiment A, you apply dual lobe editing pulse for GABA and glutathione editing simultaneously. So the question comes in, how do we get GABA and glutathione edited spectra from this method? So again, you repeat the same steps you did for megapress. For glutathione, you take all the ons minus all the offs for glutathione. And for GABA, again, you take all the ons minus all the offs. And this is where you apply the Hadamard reconstruction. Hadamard reconstruction involving A minus B plus C minus D gives you glutathione edited spectrum. Reconstruction involving A plus B minus C minus D gives you GABA edited spectrum. And now Hadamard encoding and reconstruction gives the technique its name, Hadamard encoding and reconstruction of mega edited spectroscopy, or in short, HERMES. So let's look at it. How does it affect your conventional spectrum? Starting from experiment D, you're not applying any editing pulse. You end up getting a conventional MR spectrum. In experiment C, you apply editing pulse at 4.56 ppm. You end up saturating your water signal. In experiment B, you apply editing pulse at 1.9 ppm. You end up saturating your NA signal. In experiment A, you apply dual lobe editing pulse, that is editing both GABA and glutathione. However, it also saturates your NA signal and water signal. And this is an important step to verify that your sequence works. At least you need to know that where the editing pulse is applied and whether it works the way it's supposed to work. So how do we get GABA and glutathione edited spectra? You perform Hadamard reconstruction involving A plus C minus B minus D gives you glutathione edited spectrum. GABA, get it from A plus B minus C minus D. There's negligible levels of crosstalk, as you can see, and that is because the two columns of acquisition are orthogonal to each other and they follow the columns of Hadamard matrix. So this was applied in 10 healthy volunteers, and now we have mega edited spectra overlapped with HERMES edited spectra. Mega edited spectra is acquired in 20 minutes. However, HERMES is acquired in 11 minutes. And you can see in terms of signal pattern line width, the two edited spectra agree with each other. And therefore, 22 minutes worth of megapress data can be acquired in 11 minutes without loss in spectral quality. And therefore, HERMES allows twofold increase in data acquisition rate at 3 Tesla. So we can perform this at 3 Tesla. Question is, can we perform this at 7 Tesla? Performing editing at 7 Tesla has a lot of a couple of benefits. One of them is that you get a higher signal-to-noise ratio data, and the second is that the pulse selectivity is better. So one way to reduce your macromolecule contribution is that you can perform Henry's method, or you can go higher field scanners, and because of the selectivity remains the same, but the dispersion and frequency increases, that reduces your macromolecular contribution in your GABA editing. But there are challenges to perform at spectroscopy at 70. The first one is the B1 inhomogeneity. That leads to poor voxel localization, signal losses, and the other challenge is that you can't use the refocusing pulses that you use at 3 Tesla, such as Mao pulses, because of the lower bandwidth, and that increases your chemical shift displacement error. In case of J difference editing, that reduces your editing efficiency. So several studies would conduct spectroscopy, single voxel spectroscopy, using semi-laser sequence. And how do you get that sequence? You start with the slice selective excitation pulse with the frequency modulation, and use adiabatic pulses, which are also slice selective, and with the frequency modulation. This is where the benefit comes in. This sequence implements adiabatic pulses. They are known to have higher bandwidth, so they reduce your chemical shift displacement error and increase your editing efficiency, but they're also less sensitive to B1 homogeneity. So you need to reach certain threshold, and after that, if it varies, it doesn't matter, because it just requires that minimum amount of B1 to work optimally. If you include your editing pulses, now you can perform your mega editing using single lobe, and if you include dual lobe editing pulses, you can now perform Hermes editing. So this was applied in 10 healthy volunteers, and then we have this mega edited data overlap with Hermes edited data. You can see again, just like a 3 Tesla signal in terms of signal line width, pattern and amplitude, the 2 edited spectra agree with each other. And now Hermes allows 2-fold increase in data acquisition rate at 7 Tesla. And other important point to note here is that at 3 Tesla, we perform Hermes using press localization. Over here, we used semi-laser localization. And the next step would be beneficial to have this semi-laser at 3 Tesla, so then you can gain the same benefits that that we get at 7 Tesla. So before I go into my next part of the presentation, I hope this works now. So I have a question for you, just as a recap. What are the major signals that are saturated or suppressed in some cases in the Hermes of GABA and glutathione experiment? Amazing. That means everyone's listening. Okay. All right. So let's look at the more options here. So we have NA at 2 ppm only. So again, it's important to check your sequence whether it works or not. So you look at the subspectra. So if you have a subspectra that some of them shows NA is suppressed, that means you're not performing Hermes. You're probably performing GABA editing, because you're applying the editing pulse at 1.9 ppm. That saturates your NA signal. Creatine at 3 ppm only, not sure what's happening. I think you're performing megapress experiment and you placed your editing pulses at the wrong ppm, unless you want to edit 1.9 ppm, which I wouldn't recommend. If you're saturating water at 4.6 ppm, you're probably performing megapress editing of glutathione. However, if it's saturating Na in water, not in all sub-experiments, so in one of them, and the other ones will be just Na, and the remaining ones will be just water, then that's a sign or indication that you're performing Hermes editing. Can we move? All right. So coming back to our Hermes editing. So we know that we can use Hermes for GABA and glutathione editing. Can we acquire more? So we had 2 metabolites first, GABA and glutathione, and you can see here that you can use single lobe in experiment A and B for GABA and glutathione. However, you can replace that with dual lobe editing pulses. Now, instead of performing just GABA and glutathione editing, you can perform GABA, glutathione, and ethanol editing. And then Hadamard construction involving A plus B minus C minus D gives you ethanol edited spectrum, along with your GABA and glutathione edited spectra. So this was tested in 2 healthy subjects in a mid-parietal lobe, voxel placed at sized 3 by 3 by 3 centimeter cube. So So we scanned the subjects after consuming alcohol for 1 hour, and then we broke that data down in blocks of 4 minutes that contain about 128 transients. And you can see the edited spectra gave us, as expected, 3 edited compounds. You have GABA on the left, glutathione in the middle, and ethanol on the right. And as expected, the subjects showed a rise in ethanol signal for an hour, and this sequence can be quite beneficial for studies such as studying alcoholism or alcohol use disorder, and you can break down your resolution to as high as 4 minutes, and which can give you, allow you to perform dynamic measurements of these 3 edited compounds, which previously was not possible. So we focused on single voxel editing, so that means looking at one region at a time. Can we acquire data from different regions at the same scan time? So over here we have prime sequence with very similar to press sequence. However, you replace a single band or just one slice selective excitation pulse, one lobe with dual lobe or dual band excitation, and you can perform multivoxel spectroscopy. And if you include editing pulses, you can now perform mega prime editing, or if you include dual lobe, you can perform Hermes prime. And you can use sense reconstruction to separate the signals, so you can just separate those 2 voxels of interest. And then, so working with mega prime, we acquired this data in subjects in 2 different regions, and you can see that you can get GABA edited spectrum simultaneously and in 11 minutes in 2 different regions. That allows 2 factor of acceleration relative to conventional megapress sequence. You can do this using Hermes prime as well. You get 2 edited spectra from every voxel, so that's about a factor of 4 acceleration. And this sequence can be very beneficial in studies such as tumor, where you can use one region as a control for the other. So my last part of the presentation is this is the sequence called Hercules, which is Hadamard editing resolves chemicals using linear estimation of spectra. So we perform Hermes. We got 2 or 3 edited compounds. Now, question is, can we acquire more? So we can do that using this particular editing method. If you apply editing pulse at 1.9 ppm, you can get GABA and 2-HG. Applying at 4.58 ppm, you can get glutathione, NAG. And at 4.18, you can get ascorbate, aspartate, and lactate. So how do you get these 3 edited spectra? 7 edited spectra. So you have, in different combinations, different spectra. So at some spectrum, A plus B plus C plus D gives you all the major metabolites. In A plus B minus C minus D gives you GABA edited spectrum. In A minus B plus C minus D gives you glutathione edited spectrum. Now let's look at the components of this spectra. So as expected, GABA comes from GABA edited spectrum and 2-HG as well. And now you have glutathione and glutathione edited spectrum. That's expected as well. And you have lactate as well. And now one of the novelty of this sequence is that you get NAG in one edited spectrum, and you get NAA in the sum spectrum. So you can use this information from the sum spectrum, getting an NAA signal, the quantification of NAA signal, and you can use that to precisely quantify NAG and the glutathione edited spectrum. And this is the novelty here is because at 3 Tesla, you can't really separate the 2 ppm singlet between NA and NAG, so this sequence comes in handy when you want to measure them separately. And then after that, you get aspartate, you get ascorbate, and then the rest of the major metabolites that you're interested in, such as total creatine, total choline, glutamate, and so on. So this simulated spectra, you can also use them to model your in vivo spectra using linear combination of simulated basis functions. So over here we have an in vivo data, and you can see that when fitted with this simulated spectra, you have this simulated spectra following the in vivo data really well. And now, for the first time, you can perform all the measurements that you can for all the metabolites, or almost all metabolites you can be interested in. And you can see such as ascorbate, aspartate, GABA, and glutathione, lactate, NA, NAG, and the list goes on. So the list of edited metabolites at 3 Tesla is very similar to unedited metabolites that you can get at 7 Tesla. And just to summarize my talk, so we have megapress sequence. However, it's largely focused on single metabolite editing, and we need to standardize this sequence for the reason mentioned before. That is, it allows us to conduct comparable multi-site and multi-vendor studies. It's platform independent, so you can conduct using any scanner. Faster editing has a lot of benefits. You can acquire multi-metabolite data, and it's compatible with 3 Tesla and 7 Tesla when it comes to Hermes, and it's compatible with common localization techniques such as press and semi-laser. Prime allows you to accelerate spatially, so you can acquire data from different regions at the same time. And finally, Hercules, which performs editing at a factor of more than 4 times. I'd like to thank my colleagues in and out of Hopkins, also representatives from Siemens, GE, and Canon. The standardized sequences or the universal sequences are available at all of these vendors, and you can contact me to find out more. And I would like to thank the NIH grants for supporting the study. Thank you.